So the next speaker will present, on Zoom, Wei Li from Beijing Normal University, modifications of visual cortical processing by implicit learning.
Hi, good afternoon. Can you hear me?
Yes.
Congratulations on the 60th anniversary of the Smith-Kettlewell Institute. I would like to thank Dr. Chuang Ho and the meeting organizer for inviting me. I'll take this opportunity to share with you our studies on learning-induced plasticities in visual cortical areas in macaque monkeys. The learning and memory can be classified into two large categories. One category is declarative or explicit, such as remembering some facts or events. The other category is non-declarative or implicit. The acquisition and recall of implicit memory usually don't reach our conscious awareness. So my talk will focus on two forms of implicit visual learning, namely perceptual learning and field learning. So the first part of my talk is about perceptual learning. Visual perceptual learning, that is improvement with training visual tasks, is commonly seen in some professionals, such as radiologists. Regarding the neural mechanism of perceptual learning, there has been an ongoing debate. One point of view argues for refined readout process for better decision-making in the frontal parietal areas. The other point of view argues for refined sensory representation in the visual cortex, such as V1. In fact, these changes are not mutually exclusive. Our studies have consistently shown that visual perceptual learning is mediated by multiple concerted processes. So we trained monkeys in 3 different detection tasks. One task is local orientation contrast detection or orientation singleton detection. The second task is global contour detection. And the third one is global form detection. We used the glass patterns here. So for all these 3 types of stimuli, the target saliency or the task difficulty could be parametrically controlled from very easy conditions, the popping out condition, to very hard, difficult condition. So in the first training task of orientation singleton detection, the 2 stimulus patterns were simultaneously displayed, and the target orientation singleton with different orientation contrasts against the background could appear in either of these 2 patterns. The monkey indicated the target by making a cut as fast as possible. So during the task, we recorded V1 responses using the microelectrode arrays implanted in V1. So the monkey's behavioral performance improved with training. Figure A shows the detection accuracy as a function of the orientation contrast of the singleton. Different curves indicated different training days. So for targets with small and intermediate orientation contrast, the accuracy improved with training. So panel B shows the monkey's reaction times. So for both small and large orientation contrast, there was a general reduction in the reaction time across training days. That is an increase in target detection speed. So as a pre-training baseline control and in a simple fixation task, we recorded V1 neurons responses to the targets with different orientation contrasts. So time 0 indicates stimulus onset. So the neural signals representing different orientation contrasts appear from the beginning of neural responses, indicating a feedforward or bottom-up process for detecting the orientation singleton. But after 10 days of training in the detection task, we observed a dramatic enhancement of the orientation contrast signals in the late component of neural responses. the early bottom-up signals remained largely unchanged in comparison with the pre-training control. However, these early signals, V1 signals, became more correlated with animals' behavior, as shown in the next slide. So for the same target with a small orientation contrast, so we separated the trials into two groups, the better trials and the worse trials. In better trials, the animals detected the target rapidly and successfully. But in the worse trials, the animal failed to see the target or took a long time to detect the target. So V1 responses were generally higher in the better trials than in the worse trials. So we computed the better-worse difference, the red curve minus blue one, and compared the 3 training stages during the early training days, intermediate training days, and the late training days. So we can see the better-worse difference increased in the early component of V1 responses with training and decreased in the late component of V1 responses. This suggests that training results in better utilization or readout of early V1 signals for solving the detection task. So in this task, the task relevant information is local discontinuity in orientation between the singleton and the background. So in the second training task of global contour detection, the task relevant information is global continuity in orientation between the contour elements. So the contour saliency was controlled by varying the number of collinear bars forming the contour, for example, 9 bars versus 5 bars, and by varying the spacing between the collinear bars. With a very large spacing between the collinear bars, the contour basically disappears. So in the chart, 2 stimulus patterns were displayed for 500 milliseconds, and the contour was randomly embedded in either of them. The monkey indicated the contour by making a scud. We tracked day-by-day changes in V1 using the implanted microelectrode array. So for each V1 neuron, we measured its responses to the noise pattern without any contour and to the contour pattern with a global contour. So since we have access to many neurons, here I show the 2 red circles that show 2 sample neurons with receptor fields on the contour path. And the 2 blue squares indicate 2 neurons on the background. So for each neuron, we constructed the tri-by-tri distributions of tri-by-tri firing rates in response to the noise pattern and to the contour pattern, and we computed the D prime as a measure of the neuron's contour detection ability. So in figure B, these numbers, percentage values, indicate the monkey's behavioral performance with a clear learning effect. So in each small panel, the circles and the squares indicate the receptive field centers of VM neurons relative to the contour path. So we see 2 complementary effects here. So a progressive increase in responses to neurons on the contour and progressive decrease in responses to neurons on the background. So both the contour facilitation and the background inhibition significantly contributed to enhance the contour detection ability. Based on the tri-by-tri spike counts of all recorded neurons, we used the support vector machine SVM to decode the population responses to the contour and noise patterns. So the isolated dots here show the day-by-day decoding accuracies, which are compared with the animal's behavior learning curve, the solid curve. So for this monkey 1, the decoding accuracy increased in parallel with the behavior, indicating enhanced sensory representation of the contour. But for monkey 2, the decoding accuracy also increased. But during the early training days, the decoding was much better than the behavior. suggesting that the animals are unable to use or read out the counter information encoded in V1. So such a difference was reduced with training, suggesting that the readout process was refined. So note that training only modified the late component of V1 responses. The early component remained unchanged, similar to the previous orientation singleton detection task. So the delayed counter facilitation or background suppression suggests the feedback modulation from higher order cortical areas. So in the third training task of global form detection, we simultaneously recorded neurons in V4 and ventrolateral prefrontal cortex, VLPFC. So previous studies have shown that early visual areas V1 and V2 do not encode the glass patterns. V4 is the initial stage to encode such global forms. So the VLPFC is well known to play an important role in processing stimulus features and in-mediating feature-based attention. So with these experimental settings, we could do this during the perceptual learning in both visual and association cortical areas. So in this task, the glass patterns were generated within a full screen background of random dots. The saliency of the target was controlled by varying the proportion of coherent dots forming the glass pattern, which took the radial or concentric form. So for example, for coherence 0.0, it means the glass pattern within the target area, 30% of dots are coherently organized, but 70% of dots are just random. So to examine possible stimulus specificity of the learning effect, we first trained the monkeys to detect the radial glass patterns for several weeks, and then to detect the concentric glass patterns. So by using the SVM classifier, we quantified to what extent the population responses of V4 and PFC neurons were predictive of the target location and thus was useful for solving the detection tasks. So to do that, we labeled each trial with the target location, left versus right. And the labeled trial-by-trial spike counts were used to train the SVM classifier to decode the target location. So the decoding accuracy are shown for 4 different glass pattern coherences here, showing 4 different panels. And for each training day indicated by the x-axis. So the blue and the red curves indicate the decoding results from V4 and PFC respectively. We see that for both, the decoding accuracy actually increased with training. For example, for coherence 0.6, the red curve, blue curve increased with training in parallel with animals' behavior. But actually, during the early training days, we see that the red and the blue curves almost overlapped. But with further training, the red curve decoding based on PFC surpassed that from V4, the blue curve, indicating that the readout process in PFC was improved with training. After the learning effects plateaued, we changed the configuration of the glass pattern from radial to concentric form. So we see a relearning effect. So both the decoding accuracy and the animal's behavior dropped in the beginning of the training, and the decoding accuracy and the behavior performance increased again with training. This suggests a certain degree of stimulus specificity of the learning effect. So the above analysis was based on SVM decoding of the target locations, regardless of the animal's subjective choice. It is well known that PFC conveys multiple information contents, including sensory evidence and behavioral choice. To examine whether training could affect choice-related neurosignals, we relabeled each trial with the animal's choice and conducted the SVM analysis. In this analysis, we evenly split the training days into the early days and late days for radio glass pattern training and for concentric glass pattern training respectively. So a total of about 2 months. So actually, we isolated only those trials in which the target was absent. There was no glass pattern at all. So in some of these trials, the monkey selected the lower left quadrant, and in the other trials, the monkey selected the upper right quadrant. So in this case, the decoding accuracy entirely reflects the animal's behavior choice. So we can see that the y-axis indicates decoding accuracy, and the time x-axis indicates the time after stimulus onset. So we can see the choice-related signals increased in both PFC and in V4 after the training. In addition, actually, the choice-related signals emerged earlier in PFC than in V4. The red curve leads the blue one, suggesting a backward propagation of the signals. So by controlling for the choice-related signals, we could further dissociate the neural signals specific to the glass patterns, that is the target-related signals. So in this case, we compared two conditions in which the target was present versus the target absent, but the animals always selected the same location. With this, in this case, the choice-related signals were completely removed. So based on V4 and the PFC responses, we used the SVM decode these two stimulus conditions. In both V4 and PFC, the target-related signals increased with the glass pattern coherence. For example, the upper row, early training days for 3 different GP coherences. So if we compare the early training days, the upper row, with the late training days, the bottom row, for the same GP coherence, we can see learning-induced enhancement of the target-related signals. Note that the target-related signals emerged earlier in V4, the blue one, than the target-related signals in PFC, the red curve. So here's a summary of visual perceptual learning. Visual perceptual training results in concerted changes in sensory representations, top-down influences, and the readout processes. The most pronounced changes in the visual cortex are seen in the late components of neural responses mediated by inter-area interactions. So another form of implicit learning is field conditioning. So unlike visual perceptual learning, field learning can rapidly modify the early components of V1 responses. Before the conditioning experiment, we measured neural responses in V1 as a function of greeting orientations. Here is the typical orientation tuning curve. So this condition was used as a preconditioned baseline control. Figure B shows the conditioning paradigm. Drifting greetings tilted to one side, for example, the leftward tilted greetings, was used as a conditioned stimulus, the CS, which was paired with very strong air puff delivered to the face at the end of the trial. So the greetings tilted to the other side was termed the non-conditioned or neutral stimulus. And during the delay period after the stimulus greeting display, and before the delivery of the air puff, the unconditioned stimulus, we tracked the animal's eye blink reflex. So we can see the eye blinking frequency was much higher in the CS condition than in the NS condition, suggesting the establishment of the conditioning effect. So we compared the V1 responses in the conditioning session with those in the preconditioning control experiment. So compared with the pre-training baseline control, the blue curve, we observed a significant enhancement in V1 responses to the CS greetings, but not to the NS greetings, non-conditioned greetings. So figure B shows the results from reversal learning. That is, within the same conditioning session, the CS and NS greeting orientations were switched back and forth in different blocks of trials. The two vertical dashed lines R1, R2 indicate two reversals. So the x-axis indicates the sequence of the CS presentations, which were randomly interleaved with the NS. So after the CS-NS switch, the field-related signals in V1 also reversed. So the orientation tuning curves of V1 neurons were also modified by field conditioning. So we separated V1 neurons into three groups according to their preferred orientations relative to the CS orientation, the LPAF associated orientation. So the left panel shows neurons with preferred orientations centered around the CS orientation. So we see an enhancement of the tuning peak. The middle panel shows neurons with preferred orientations roughly orthogonal to the CS orientation, and then we see an elevation of the tuning flanks. So for the remaining neurons, the CS orientations was on the flank of the tuning curves. So we see an asymmetric elevation of the tuning curve. So the conditioning effect was specific not only to the conditioned greeting orientation, but also to the conditioned visual field location. The early field-related signals were absent at the unconditioned visual field locations. So the field learning effect seen in V1 seemed to be subliminal. So when the CS and NS greetings are superimposed with horizontal drifting greetings, the subjective percept becomes moving plates in which the CS and NS orientations are not clearly visible, but the plates containing the CS component induced the field-related signals in V1 similar to the isolated CS greetings, but the plates containing the NS component did not induce any field-related signals. So the summary of the field learning, unlike perceptual learning, field learning induces changes in V1 from the outset of neural responses, suggesting modifications to the feedforward process for proactively tagging their inputs that are predictive of imminent threat. So to conclude, recurring real experience can shape different cortical processes. Notably, V1 is capable of conveying multiple task and behavior relevant information from these experiences. So I would like to thank my lab members and also my collaborators. Thank you. I'm ready to take the question.
Thank you so much. They're really remarkable results. We'll have time only again for one brief question while the next speaker is set. So, any questions? Yeah, no, I don't. Are there questions to the speaker? I have a question. You, in relation to fear conditioning, you were explaining the results with change in the feedback, in the feed-forward processing for proactive tagging based on predictive visual stimulus information. This is kind of, I'd like you to elaborate, but I'm not sure how much time you have now. Is it possible to give some short answer, or should we leave this for the general discussion? So because you have feed forward, you have for proactive, but it's based on predictive form of coding. So in other words, the predictive part usually seems to require feedback, not only feed forward. So it's affecting the feed forward, but what is the role? How is this mechanism implemented? How is this the mechanism implementing this change in the feed forward process?
This is a great question, but due to time limit, I just give a brief answer. Actually, V1 response latency is about 40 milliseconds. So field-related signals basically start from the first spike, but the neurons in amygdala respond to real input with a latency about 100 milliseconds. So our speculation is that during the field conditioning process, there's an interaction between the amygdala and the neural cortex, in our case, V1. So during the conditioning, somehow the feed forward connection could be quickly modified due to, for example, synaptic plasticity as expanded by Merganka. So after establishment of the field conditioning, so the real inputs could activate such kind of field memory already installed in V1. So we could further discuss the feed-forward feedback at the end of the session. Thank you.
Okay, thank you so much.